Okay, let's start with today's paper. So question 1 says that the olive plant, Olea europaea, is grown in many parts of the world. The fruits of the plant, which is the olives, and the oil that can be obtained from the fruits, which is olive oil, provide food for humans. Now, triglycerides are the main type of lipid in olive oil. They are synthesized in the olive plant from glycerol and fatty acids. Now, scientists can analyze samples of different olive oils to identify certain things. These include the fatty acids used to synthesize triglycerides, as well as the composition of the different triglycerides that are present. Now in A, they say that table 1.1 shows some details of the 5 most common fatty acids found in samples of olive oil produced by olive plants grown in different regions in Portugal. Now they give us a key here. We have CD, which is a ratio of the number of carbon atoms to the number of double bonds in the hydrocarbon chain. And then X is some missing detail. Let's have a look at how triglycerides are formed and how their composition might differ. Now, triglycerides are formed through a reaction where an alcohol, which is called a glycerol, bonds with a carboxylic acid, which is the fatty acids. Now, 3 new bonds will form between the glycerol molecule and the fatty acid molecules. When these bonds form, water is released, which means that this is a condensation reaction, and the reverse of this reaction will be a hydrolysis reaction. These fatty acids can differ in composition in many ways. One of the ways is the fact that they have different lengths of hydrocarbon chains. So one might be 23 carbons long and another one might be 16 carbons long. Secondly, they can either be saturated or unsaturated. Now, saturated refers to a carbon chain that has no double bonds. So it has the maximum amount of hydrogen bonds to the chain. Unsaturated means that there is double bonds present. Now, when we look at unsaturated fatty acids, they can either be monounsaturated or polyunsaturated. So mono means one, so it means that it only has one double bond, and poly means more than one, so it has more than one double bond. Let's quickly have a look at our table and exactly what it shows us. So the first column shows the name of the fatty acid, for instance, oleic acid. Then it shows the percentage of total fatty acid content. And then it gives us our ratio between our carbon atoms and the double bonds. Lastly, they give us our chemical structure. So here we can count 1 and then the 7 multiplies with this carbon. So it's 8 plus that 1 is 9 plus that 1 is 10 plus that 7 gives me 17. But C here is 18. So I know X needs to contain a C atom. Now, if we remember from our video, we saw that fatty acids are carboxylic acids, which means that an X compound would be a carboxyl group, so COOH. Our first question then says that table 1.1 shows the CD values for oleic acid. In table 1.1, write the values for D for each of the 4 other fatty acids listed. We can see that here for oleic acid, there's 1 double bond in the chemical structure and there's 1 as a value for D. So for palmitic acid, we can't see any double bonds, so the value would be 0. For linoleic acid, we can see 2 double bonds, so the value would be 2. And for stearic acid, there's also no double bonds, so it's also 0. Lastly, we have palmitoic acid, which has 1 double bond. We then have the second question, where they say in the first column of table 1.1, draw a circle around each of the fatty acids that can be described as saturated. So remember, saturated means that it has no double bonds. Now for oleic acid, it has one double bond, so it's unsaturated. But palmitic acid would be saturated, as well as stearic acid will be saturated, as linoleic acid and palmitoic acid do have double bonds. Lastly, they ask us to state the detail of chemical structure represented by X, which is missing from table 1.1. So we already know that a fatty acid is a carboxylic acid, so this would be COOH. We can then move on to the next page. So here we have question 4, which says that the analysis of the triglycerides present in the different samples of the olive oil showed that, firstly, there are many different triglycerides present in olive oil. Each olive oil is different in its composition, but the same few triglycerides are present in all olive oils. Now they want you to, with reference to table 1.1 and to the structure of triglyceride, suggest an explanation for these observations. So you need to explain why there are only a few triglycerides present in olive oil, but they have different compositions overall. Now we can say that each triglyceride has 3 fatty acids. Now these fatty acids can be saturated like palmitic acid or unsaturated like oleic acid. The triglycerides can also have different lengths of hydrocarbon, like stearic acid that has 18 carbons in its chain and palmitic acid that has 16 carbon in its chain. Question B then says that glycerol is soluble in water. Triglycerides are insoluble in water. We have to explain why water is a good solvent for some substances such as glycerol, but is a poor solvent for substances such as triglycerides. So before we answer this question, let's quickly look at how water acts as a solvent. Now, water molecule is made up of an oxygen atom as well as 2 hydrogen atoms. Now, the oxygen atom will be slightly negative charged, and the hydrogen atoms will be slightly positive charged. This means that water is a polar molecule. Now, if we look at the structure of glycerol, we can also see that it has oxygen and hydrogen atoms attached to the ends of it. Now, the slightly positively charged hydrogen on glycerol will interact with the slightly negative charge of the oxygen on water. Because these 2 atoms have opposite charges, they will attract each other and they will form a hydrogen bond. These bonds make it possible for molecules like glycerol, which is also polar, to dissolve into water. Now, triglycerides do not have these hydrogen-oxygen bonds because they are connected to fatty acids, which means that they are nonpolar and will not be able to form bonds with water, so they will not be able to dissolve into water. We can now answer the question. So we can first say that water is a polar molecule and that it's able to form hydrogen bonds with other polar substances like glycerol, but it will not be able to form bonds with non-polar substances like triglycerides. Next, we have question C that says phloem is the plant tissue responsible for the transport of organic substances such as fatty acids from one area of a plant to another. The tissue is composed of more than one type of cell. Name the type of cell that forms the transport vessels of phloem tissue. Let's quickly look at the structure of phloem tissue. So firstly, we have the part that would transport the substances, like for instance sucrose and fatty acids. And this is a semi-hollow tube which would have cytoplasm in it, as well as sieve plates that are perforated in between each cell. So this enables phloem to transport substances in multiple directions, not just in one direction like in xylem. Now this part of phloem is called the sieve tube element. Now adjacent to this sieve tube element are also living cells called companion cells. Now the companion cells are able to serve as the sieve tube element, especially the cytoplasm, because it has living organelles in it. It will have a nucleus and mitochondria and endoplasmic reticulum. There's also some fenestrations between the sieve tube element and the companion cells that makes movement of molecules between the two cells possible. Companion cells also very important for the unloading and uploading of sucrose into the sieve tube element. So we can answer question C as the sieve tube element. We can then go to the next page, and here we have question 2. So they say that people who become infected with human immunodeficiency virus or HIV are at risk of developing HIV or AIDS, particularly if antiretroviral therapy is not available. In people infected with HIV, the use of ART also helps to reduce transmission of the virus to uninfected people. They then ask us to outline 2 control methods other than ART that can be used to reduce the transmission of HIV. Now we all know that the first way to reduce the transmission of HIV is by practicing safe sex. Screening of blood should also occur for transfusions, and drug users should avoid sharing needles. Now, mothers that are infected with the HIV virus should avoid breastfeeding as well. They then say that in people with HIV/AIDS, a serious lung disease known as pneumocystis pneumonia can result from infection by an opportunistic pathogen known as pneumocystis jirovecii. Figure 2.1 shows these cells in one stage of their life cycle as seen using a light microscope at a magnification of 600 times. They then ask us to define magnification. Now magnification is the number of times larger an image is than the actual. On the next page we have question C, which says that figure 2.1 shows that P. jirovecii is an unicellular organism, although the cells of many species of bacteria are the same size as those of P. jirovecii. Research concluded that the organism is a eukaryote and not a bacterium. In 1988, analysis of ribosomal RNA or rRNA resulted in P. jirovecii being classified as a fungus. Now the first question says that studies of the structure of P. jirovecii have identified that the cell wall is made up of polysaccharides such as chitin and 1,3-beta-D-glucan. Explain why these features helped scientists to confirm that P. jirovecii is not a bacterium. We can now have a look at the differences between a bacterium and a fungal cell. Now both bacteria and fungi have a cell wall. The difference is that bacterial cell walls are made up of peptidoglycan and fungal cell walls are made up of chitin. Now both also have a cell membrane with cytoplasm. Fungi would have a nucleus as it's a eukaryote, but bacteria will not have a nucleus. It will have a circular piece of DNA as well as plasmids. This is because it is classified as a prokaryote. Because fungi is a eukaryote, it will also have other membrane-bound organelles like endoplasmic reticulums, mitochondria, and even Golgi apparatus. Now, bacteria, being a prokaryote, won't have any membrane-bound organelles, but it will contain ribosomes to manufacture proteins. These ribosomes will also be present in fungi. So the largest difference between bacteria and fungi is the fact that bacteria is a prokaryote, whereas fungi is a eukaryote. We can then go back to the question and state that because bacteria have cell walls made of peptidoglycan, it means that P. jirovecii is not a bacterium, because remember, this organism had chitin cell walls, not peptidoglycan. The second question then says that scientists have identified other features of the cell structure of P. jirovecii. Some of these are listed in Table 2.1. They want us to complete the table by making sure that there's 4 structural features that are identified in the fungus, as well as 1 function for each structure, and whether the structural feature is present or absent in bacterial cells. So, for the structures at the bottom, there's an opening. So we know that the function of this structure is aerobic respiration. So the structure would be mitochondria. Then for functions, the smooth endoplasmic reticulum has a function to synthesize lipids. Now ribosomes will be present in bacterial cells, but the smooth endoplasmic reticulum, Golgi body, as well as mitochondria will be absent from bacterial cells, as bacterial cells can't have any membrane-bound organelles. On the next page, we have question D that says P. jirovecii can adhere or attach to squamish epithelial cells of the alveoli and to the network of fibrous proteins that support the alveolar wall, known as the extracellular matrix. Example of proteins in the extracellular matrix are elastin as well as collagen. Now, adhesion or attachment of the fungus to alveolar epithelial cells and the extracellular matrix stimulates the growth of its population. Now, the cell surface glycoproteins known as GPA glycoproteins are essential in allowing the fungus cells to adhere to alveolar epithelial cells and the extracellular matrix proteins. They want us to suggest how a GPA glycoprotein is able to adhere to alveolar epithelial cells and the extracellular matrix protein. So let's have a look at how all this is possible. Now, our body cells have receptor proteins on their cell surface membrane. These receptor proteins have a specific shape. It also works as an identifier molecule of the cell. Now, the fungal cell will have the GPA proteins. These GPA proteins will have a specific shape that will be complementary to the receptor proteins on our cells. This means that they are able to attach and bind to our alveolar cells. So we can answer this question by saying the GPA glycoprotein will bind to receptor proteins on the cell surface of the alveolar cell or to extracellular matrix proteins. Now, this is possible because the GPA glycoprotein has a complementary shape to these proteins. We then have the second question that says one consequence of the pneumonia that results from P. jirovecii infection is a decrease in the quantity of oxygen that's delivered to body tissues. Explain why a severe infection results in a decrease in the quantity of oxygen that's delivered to body tissues. Now, we have to remember that these fungal cells are attaching to our alveoli that are supposed to be in close proximity to our capillaries for gas exchange. So we can say that the alveolar cells are surrounded by the fungal cells, and this decreases the ability of oxygen to diffuse between the alveolus and the capillaries. We can also say that it will restrict the alveoli to stretch and recoil, as it will impair the elastic fibers found in the extracellular matrix. On the next page, we have question E. So here they say that the fungus produces an enzyme known as 1,3-beta-D-glucan synthase. The enzyme catalyzes the synthesis of 1,3-beta-D-glucan. Now remember, this glucan is found in the cell wall of the fungus. Now the therapeutic drug caspofungin is a non-competitive inhibitor of 1,3-beta-D-glucan synthase. So before we go on with this question, let's have a look at the difference between a competitive and a non-competitive inhibitor. Now, enzymes can encounter two types of inhibitors. If we have our enzyme molecule here, we have two attachment points. We have the active site, and this is where our substrate will be binding for the catalyzing of the reaction. And then we also have our alternative binding site. Now in competitive inhibitors, our inhibitor will attach to the active site. This means that the substrate won't be able to attach there. But it also means that the competitive inhibitor and the substrate will compete to bind to the active site. So one can always outcompete the other. Now, a non-competitive inhibitor will bind to the alternative binding site. This will cause the active shape to change its shape, and the substrate won't be able to fit in the active site at all. This means that even though I increase the substrate concentration, it won't be able to outcompete my non-competitive inhibitor. Let's go back to my question. They say that with reference to the mechanism of action of caspofungin, remember this is a non-competitive inhibitor, explain how the drug may be useful to treat cases of pneumonia caused by the fungus. So we can firstly say that the drug will bind to a site on the enzyme other than the active site. We can also say that this will change the shape of the active site. So this means that the substrate will not be able to fit into the active site. So this will lead to less or even no product to be synthesized. Now remember, the product here was 1,3-beta-D-glucan that was used in the cell walls. So if it's not being synthesized, it will weaken the cell wall of the fungus, which can eventually lead to lysis of the cell. On the next page, we have question 3 that says, during transcription, base pairing occurs between nucleotides. Now, figure 3.1 is a diagram to show complementary base pairing between a DNA nucleotide and an RNA nucleotide. Only the base pair is shown in the molecular detail. So before we go to question A, let's go look at the whole molecule in more detail. So let's first have a look at the different bases that we can get. Now there are two main groups. The first group is purines, and they're made up of two cyclic rings. The first one is adenine, and the second one is guanine. Now the second group of bases that we get is pyrimidines, and they're only made up of one ring structure. So here we get thymine, cytosine, and uracil. Now remember, uracil is only found in RNA, whereas thymine is only found in DNA, but adenine, guanine, and cytosine are found in both. So let's look at the structure of how the DNA strand would bind to the RNA strand. So on my DNA strand, I'll have a phosphate connected to a sugar that's then connected to my nitrogenous base, which is my base pairs, adenine, guanine, thymine, or cytosine. On the RNA strand, I'll have my base pairs, which is adenine, guanine, cytosine, or uracil, which is then connected to a sugar as well as a phosphate. Now, between the two base pairs, we can find hydrogen bonds that keep them together. And then where the phosphate group connects to the sugar group, we can find the phosphodiester bond. And we can see it clearly if we add another nucleotide or sugar to our string. So let's have a look at the first question. They ask us to explain why figure 3.1 does not include any phosphodiester bonds. And that is because phosphodiester bonds form between the nucleotides on the same chain, and only one base is shown per strand in figure 3.1. Question B then asks to identify and describe the DNA-RNA nucleotide pairs shown in figure 3.1. You may add labels and annotate figure 3.1 if you wish. So I found it easier to actually give my answer on figure 3.1 for this question. So the base on the left-hand side, or on my DNA strand, would be guanine, which means that on my RNA strand, my nucleotide would be cytosine. There would also be hydrogen bonds in between holding the bases together. We also know that cytosine is an example of a pyrimidine, and guanine is an example of a purine. We can also see on the right hand side they already labeled the sugar as a ribose sugar, which means that on the left hand side would need to be a deoxyribose sugar. This shows us that the right hand side is my RNA chain and the left hand side would be my DNA chain. Next we have question 4, which says that adult stem cells are undifferentiated cells that are found in most animal tissues. Now, adult stem cells can divide by mitosis throughout their lifespan to form identical stem cells, which are called self-renewal. Or they can form cells that can differentiate into the functioning cells of that tissue. Now, mitosis is important for the repair of tissues. They then ask us to explain what is meant by the repair of tissues. Now, this is the replacing of cells that are damaged. Question B then says that uncontrolled cell division is a characteristic feature of tumor formation from a differentiated cell. Describe other features of tumor formation from a fully differentiated cell. So firstly, it is the result of a mutation that can cause continuous cell divisions. Now, usually within cell division, there are checkpoints to either slow down cell division or stop cell division or check for any mistakes. Now, these checkpoints may not occur in tumor cells as in regular cell cycle cells. Now, the tumor cell will lose its original function and may spread to other tissues. Question C then says that telomeres prevent the loss of genes. Adult stem cells have chromosomes with long telomeres. Explain why long telomeres are an advantage to cells that carry out many cell cycles. So firstly, each time DNA replicates, the telomeres will shorten. Now, telomeres do not contain genes that can be lost when telomeres shorten. This will protect the DNA from becoming damaged during cell division. So on the next page, they say hematopoietic stem cells, or HSCs, are adult stem cells that are located in the bone marrow of bones. Now, these cells have a role in the formation of blood cells. Now, figure 4.1 is an outline summary showing the formation of some of the different types of blood cells that can be formed from HSCs. The first stage is the division of HSCs to produce progenitor cells. These cells are also able to divide by mitosis, but are not stem cells. So before we go on to question D, let's quickly look at this diagram in a bit more detail. So here we can see our HSCs that can undergo self-renewal by replicating into a cell that is themselves. They can then also differentiate into CMP cells. These CMP cells can then become MEP cells that can either differentiate into red blood cells, which is responsible for the transport of oxygen, or platelets, which is responsible for blood clotting. Now, instead of turning into MEP cells, the CMP cells can become GMP cells. Now, these are basically our phagocytes. So these can become mature neutrophils, which are usually smaller in size, or monocytes. Now, monocytes is the name we give our cells if they are found in the blood, and when they're found in our organs, we give them the name macrophages. So the HSCs can also become CLP cells that will become our B lymphocytes and our T lymphocytes. We can also see that our mature B lymphocytes become a certain cell when an immune response is triggered. Now these are called plasma cells, and plasma cells are able to secrete antibodies. Let's get back to our question. They say, with reference to figure 4.1, explain why GMP cells, which are progenitor cells, cannot be described as HSC cells. So remember, here is our GMP cells. They can only differentiate into neutrophils and monocytes. So they cannot be described as HSC cells because they have already started to differentiate. They will only be able to form mature neutrophils and monocytes, and not all type of blood cells. They are also not able to undergo self-renewal like HSC cells. So on the next page we have question E, that states that figure 4.1 shows that monocytes differentiate into cell types X, which has a similar function to neutrophils. They want us now to name cell type X. So if we go back to our figure, we can see cell type X here. So it is differentiated from monocytes, and that would be called macrophages. So we can answer the question just by saying macrophages. We then have question A, that says cell type Y, shown in figure 4.1, releases molecules with antigen binding sites. Name the molecules released by cell type Y. Now we know that these molecules that have the antigen binding sites are known as antibodies. We then have question G. So they say that the differentiation of T lymphocytes begins in the bone marrow and continues in an organ known as the thymus to produce fully differentiated T helper and T killer cells. So in the thymus, T lymphocytes that bind to self-antigens are destroyed. Explain why T lymphocytes that bind to self-antigens need to be destroyed in the thymus. So let's quickly think about it. If we have an antigen binding site on our T lymphocyte that has a specific shape, it should bind to an antigen that has a complementary shape. When binding to the antigen, it will signal the body or itself to destroy the antigen that is either a protein of a pathogen or a pathogen itself. But if it has an antigen binding site that's complementary to a self-antigen, meaning it's complementary to a protein found on the body cells inside of our body, then when it's exposed to our body cells, it will attach to our body cells and it will activate an immune response that will attack our body cells and start destroying our body cells. So we can answer this question that if these cells are exposed to self-antigens, it will destroy these cells. These cells are destroyed so that only T lymphocytes that can attach to pathogens are released into our bloodstream. Question 5 then says that malaria is an infectious disease caused by the protist Plasmodium. As part of its life cycle, Plasmodium infects the human red blood cells. Researchers can compare hemoglobin from the red blood cells of a healthy person with hemoglobin from a person with malaria. Now, throughout the world, most deaths from malaria are caused by Plasmodium vivax and Plasmodium falciparum. They then ask us to name one other species of Plasmodium that causes malaria. And this can be either Plasmodium ovale or Plasmodium malaria. Question B then says that in the laboratory, oxygen at different partial pressures can be bubbled through a solution of hemoglobin to determine the percentage saturation of hemoglobin at each partial pressure. A graph constructed from the results is known as an oxygen dissociation curve. They then give us figure 5.1, which is an oxygen dissociation curve for normal adult hemoglobin in humans. So this is for normal humans. This will be our oxygen dissociation curve. Now we can see that it gives us the percentage saturation of hemoglobin with oxygen against the partial pressure of oxygen. And we can also see that it kind of has an S shape to it. On the next page, we have the first question that says, in the experiment used to obtain the results shown in figure 5.1, the temperature and pH was standardized. Explain what the researchers would consider when deciding which temperature and pH to use in the experiment. So firstly, the pH which would need to be used should be approximately 7.4. as this is the pH of our blood. Now the temperature would need to be approximately 37 degrees Celsius, as this is the normal body temperature for humans. The second question then says, using a different, more rapid technique, researchers compared the hemoglobin contained in red blood cells of a healthy person with the hemoglobin of a person with malaria who had been infected with P. vivax. By analyzing the results, the researchers concluded that the oxygen dissociation curve of a person with malaria would be shifted to the right. Let's quickly look at our graph and see how this would look. So shifted to the right is in that direction. So our graph would look something like this. This means that if we take a certain percentage of saturation, which is 20, for our normal person, it would be obtained at 2 kilopascals of partial pressure. But if we take the same percentage saturation for a person with malaria, it will only be obtained at approximately 4 kilopascals of pressure, meaning that I need a higher partial pressure for the same percentage saturation of oxygen. Let's read our question. They say, with reference to figure 5.1, explain how a shift to the right of the oxygen dissociation curve would affect oxygen loading in the lungs and unloading in respiring tissues in a person with malaria. So firstly, we can say that the affinity of hemoglobin for oxygen will decrease. Thus, a lower percentage saturation of hemoglobin with oxygen will be achieved. This means that it will be more difficult to load oxygen in the lungs, as a higher partial pressure is needed. We can also say that oxygen will be more readily released in respiring tissues. On the next page, we have question C. And here they say a red blood cell that is infected with Plasmodium cannot carry out its function as effectively as a normal red blood cell. They then want us to describe how the size and the structure of a red blood cell is related to its function, other than the fact that it contains a very large number of hemoglobin molecules. So we can firstly talk about the size of the red blood cell. It has a diameter of approximately 6 to 8 micrometers, and this makes it possible for it to pass through our very small capillaries. It also has no nucleus or mitochondria, which leaves more space for hemoglobin. but is also a concave shape, which increases the surface area for the uptake of oxygen. This also reduces the distance for diffusion. On the next page, we have question 6, our last question. So here they say that the transport of water from the soil solution to the xylem of roots occurs by the apoplast and symplast pathway. Now, mineral ions can be transported dissolved in water. Describe the transport of water from the soil solution to the endodermis of roots by the apoplast pathway, and explain why this pathway cannot continue at the endodermis. So just a quick recap, remember the symplast pathway is through the cytoplasm as well as the vacuoles, whereas the apoplast pathway is through intercellular spaces. So we can answer this question by saying water will move through the intercellular spaces by osmosis down a water potential gradient. The endodermis is made from suberin, which is a waxy waterproof substance. Thus, water will not be able to pass through the Casparian strip. Then question B says that researchers investigated the mechanism of transport used for the uptake of potassium ions into root epidermal cells at different concentration of potassium in the soil solution. Complete table 6.1 to provide information about the two different transport mechanisms that were identified by the researchers. Now remember, potassium cannot just move by diffusion alone. So the first net movement says that it's against the concentration gradient. This will be an example of active transport, which means that yes, membrane proteins will be needed, and yes, ATP will be used as well. The second one says down the concentration gradient. Well, we already know that it cannot just move by simple diffusion. So this must be facilitated diffusion. So with facilitated diffusion, membrane proteins are also needed, but ATP is not used.